everyone, I'm Jennifer, and I'm excited to be here today to talk about our method called BKinD-3D for doing self-supervised 3D keypoint discovery for multi-view videos. The goal of our method is to discover 3D keypoints from videos of moving agents without requiring any 2D or 3D annotations. In comparison, previous methods would require 2D or 3D annotations during training. Our method has been applied to discover keypoints on both humans as well as rats. The intuition of our method is that we start from 2 frames of a behavioral video, which is a video with stationary camera and background with moving agents. And we want to discover these key points in a self-supervised way that can reconstruct spatiotemporal difference of the moving agent. The idea of BKinD-3D is that we can now apply the spatiotemporal difference reconstruction laws across different views. So encoding information across views into a common volumetric bottleneck and doing spatiotemporal difference reconstruction now across views. Our method is motivated by the importance of doing behavior analysis in 3D, which is not only important for a variety of model organisms. For example, in scientific experiments, this type of 3D key points can be used to correlate with different types of neural activity to better understand the neural basis of behavior, as well as performing pharmacological evaluations. For humans, there are a variety of different user applications, which involve, for example, human-robot interactions, as well as healthcare monitoring. In both of these cases, understanding the 3D movement of these organisms can be very helpful for these downstream tasks. For related areas, of course, one important related area to 3D keypoint discovery is supervised 3D pose estimation. We found that, of course, in this area, these models require 3D annotations for training. And these annotations can be very expensive to obtain, either through motion capture or human hand annotations. Another area of work performs 3D from 2D, for example, through lifting models. However, these models require either 2D or 3D annotations for training. For 3D keypoint discovery, another related area is, of course, 2D keypoint discovery. And previous methods in 2D keypoint discovery have often used visual data, often from a single view, to discover 2D keypoints or skeletons of these organisms. Our work aims to do 3D keypoint discovery without requiring either 2D or 3D annotations. So now, as compared to 2D keypoint discovery, our method leverages information across multiple different views in order to now discover 3D keypoints instead of 2D keypoints. Next, I will pass on to Emil to explain to you the details of our method, various results, as well as the conclusion.
Thanks, Jennifer. Hi, I'm Emil. Our BKinD-3D method relies on a spatiotemporal difference. The intuition behind this is that spatiotemporal difference provides a strong cue for motion. Ultimately, we want the key points to be capturing motion. Why is that? Consider the case where we have a pair of scissors. A human would probably put key points at these particular places. But why? If we then open the scissors, we can see that these key points actually capture much of the motion of the pair of scissors. Additionally, the motion of particular key points in relation to other key points provides a cue for connectivity. This is the background that we rely on for BKinD-3D. We start with a frame at I sub t and a future frame I sub t plus k, which is then fed through a shared encoder. Then these appearance features are fed through a decoder in order to obtain 2D heat maps, which encode the 2D key points. We then repeat this across all the camera views to obtain multiple 2D heat maps. We then aggregate these 2D heat maps into a 3D volumetric bottleneck, which is then processed with a V2V volume-to-volume network. Next, we draw differentiable edges between every pair of points. This is then projected onto each camera view using the camera projection matrices. The projected edges are then fed into a reconstruction decoder along with appearance features in order to do the spatiotemporal difference reconstruction. We repeat this for all views. Since the edges are differentiable, we're learning weights where we can actually learn to ignore or incorporate certain edges. So for instance, we can learn that certain edges will be kept. And from here, we can get our connectivity graph for the agent. We also have a learned length constraint where we are enforcing or trying to encourage that the edges are a constant length through time. Additionally, in order to prevent key points from converging onto one another, we use a separation loss. Quantitatively, our BKinD-3D method significantly closes the gap with other supervised methods as well on human 3.6 as well as rat 7M. Next, we present qualitative visualizations. As we can see, BKinD-3D, especially with 30 key points, captures the structure of the agent very similar to the ground truth. However, we do not have any ground truth labels. Next, we investigate one particular application of the 3D key points, in particular, human action recognition. We train a simple CNN classifier in order to predict human action labels. We train this on top of 3D key points using ground truth labels as well as those obtained through BKinD-3D. We train and evaluate using the human 3.6 million data set, which has 16 actions. With human annotated key points, we get 64.8% top 5 accuracy, whereas with BKinD-3D, we get 64.9%. So what this suggests is that we actually don't need to spend time labeling a lot of data in order to obtain similar performance. So we can significantly ease the burden of key point annotation in applications. There are a variety of other applications where we can use BKinD-3D and 3D key points. For instance, scientific experiments where we're doing behavioral analysis or other user applications. Thank you for listening.